Hi everyone. Sorry I couldn't be there today. I accidentally double booked myself because I'm really smart. I'm here to talk to you about my experience with the marine science minor as well as my study abroad experience. As Alex has probably talked to you, you can substitute some courses with the marine science minor. And I did that a lot, as you can tell here. So what we've got here, we've got the entrance to minor. Everyone has to take that as a part of the marine science minor. I also did a study abroad I'll be talking about momentarily, which was the Bio 499A, Tropical Field Ecology in Costa Rica, and then Symbiosis, Bio 406, a very good, I would say, I took it towards the end of my career here at Penn State, but I think that would be a very good starter 400 for anyone looking in the biology field for some 400 level classes to fill out those requirements. Now, in terms of non-listed courses, I went off the beaten path quite a bit. The ones that are probably the most applicable are the two 497 classes I took. Now, if you've not taken a 497 class before, 497s are essentially a class that's offered infrequently here, like a special interest class, and the first one I took was the Comparative Invertebrate Physiology. That's a great class. It's offered every 2 years. It's mostly lab work, and you get to do your own experimental designs with live trials, and it's really awesome. Now, the other one, Ecology and Evolution of Coral Reefs, that's with the same professor I'm taking for Symbiosis. That's a great little special interest course as well. That I'm taking right now. I think that's also offered every 2 years. These here at the bottom, BMB 205S and WFS 209N. Now, these might be a bit more difficult to get approved. My advisor is Will Christensen, and he's great. And with WFS, that I think might be the WFS Intro to Minor. I was slightly interested in WFS for a while. That's Wildlife Fisheries Science. Apparently the fisheries aspect of that makes it count for marine science, so that's great. And then BMB 205S, I did this with the LEAP program, so it's not really an option that many people could carry on now, but that was phage research with a phage that impacts oysters. So that was counted, I think, between that and comparative invertebrate physiology. I already had like the lab work sort of sections of my thing covered, but the tropical field ecology also counts for that. Now, my advice is pretty general, all things considered. Really, don't be afraid to go off the beaten path here. It really worked out for me to just ask questions if anything involved marine life or water at all, and your advisor can probably make it work. Lastly, it is okay to join this minor late in the game. I joined the minor the second semester of junior year, which is pretty late to decide you want a minor. So you have time, you can make it work, and truthfully it's not that much more effort, in my opinion, because I was a biology major with the marine science minor. And I don't think it was really that much different than the classes I would have already taken, all things considered. So, really, I truthfully recommend it if you're a biology major interested in the marine sciences. Now, that study abroad experience I mentioned. I went abroad to Costa Rica to perform tropical field ecology. Did it have a lot to do with marine science? Not really, and we'll talk about that, and that's kind of why I want to talk about that. So, where'd we go? We started at a research station called La Selva. You can see the research station part of it here, but it's literally in the middle of a tropical rainforest, which is awesome. Here's our walk to class. Pretty awesome. Just over this gigantic bridge over a river. And then, once we wrapped up there, we went somewhere for New Year's Eve. This is a good time to mention that the study abroad takes place over the winter. So we left, I think, 2 days after Christmas, and we got back not even a day before class started. So it's a pretty thin little window there. So We sort of started here, went back to here for New Year's, and now we're in Palo Verde. Completely different environment here. Now, so here I am at Palo Verde, which is our dry forest, like I said. Some more examples of that. Nice and sunny all of the time. This also neighbored some swampland, and we got to see some really neat stuff here. There was a dock that we walked out onto, saw some awesome bird life. If you're a bird watcher, this trip is for you, let me tell you. The professor that runs this, Tomas, he is a big bird guy, and if you have any interest in birds at all, I think you'll get along with him, and I think you'll like the entire experience. So then we took beach day. This is the closest we got to marine science on this trip. I should probably say that now. um, this is a complete rec day. We just used it to work on projects and whatnot. I'll get into some of the research we did in a minute. I'll try not to yap your ear off too much about it. But this was the closest we got to marine science. Snorkeling is offered. It's nice. You can just chill in the water for a day. Um, so we kind of sort of bounced back here. And now we're in this place called San Gerardo, a little bit more south. This is a new research station in what's called a cloud forest. So whereas up here at Palo Verde it wasn't raining much at all, it's raining a lot here. It's very cloudy, it's very damp and muddy and moist. What a neat place this was. It was always cloudy and foggy, and it always felt like it was going to rain, even if it wasn't raining. A lot of neat stuff to see here. There was a waterfall. Hey, that's me. I know that guy. Lot of cool stuff here. But I know what you're here for. Yeah, we saw a lot of neat animals, and let me show you a few of those real quick. So that's a smoky jungle frog right there. It's a big boy. They can range in size from like that big to like that big. They're pretty cool frogs. They almost sound like a car alarm when they're getting angry with you. So they're a lot of fun. They kind of just sit outside of their hole, and if they're startled, they'll jump back in their hole. Hope you're all having a goodyful evening. Annika, did they like that joke? Alright, anyway, so these little guys are a lot like squirrels. They'll essentially take nuts and stuff, they'll dig and they'll bury it for later, and then they'll forget about it because they're really bad at remembering. So they serve a really interesting ecological niche, sort of like squirrels do here and other rodents. And they're cute. Kind of just some critter with fur. This thing is pretty neat. Not a big guy. It's like that big. This is a whiptail lizard. They're all around. I really like their blue hue to the edge of their tail there. What they'll do is they'll whip it in circles, and it'll sort of be used as a distraction for predators. The mossy stick insect. I couldn't have picked a better picture at him. I didn't realize the resolution was so low. My apologies. But this guy was really cool. It's not like the stick insects you might see around here that are all brown and gangly. This guy had like some green phalange stuff off him, and it was really cool. Strawberry poison dart frog, aka the Spider-Man frog. What a vibrant red and blue color these guys. It's only poisonous if you eat it, and they make it pretty clear to you not to eat it. So in general, I wouldn't go around eating random amphibians or putting really anything in the jungle in my mouth, unless Tomas told me to, which I did. I should clarify, I mean different little fruits and berries that he okayed. That is what I put in my... I should cut this part out of the video. Two-toed sloth. So we actually got to see both two-toed and three-toed sloths, which is really cool. He's right up here. If you can kind of see, there's his eye. And then, yeah, there he is. Really cool guy there. Love me some sloths. Red-eye tree frog. This was awesome. I was not expecting to see one because they're usually higher up in the canopy. But, this guy was super cool. I took both of these pictures. This is our TA Jack holding it. You can see his little orange toe pads here, and then if you look closely, you can see the blue stripe. Oh yeah, there's a picture of him with his eyes open. Beautiful color, just crazy animal. What a cool critter. You don't need to set alarms in Costa Rica. These guys have got it. They every morning, 5 to 5:30 in the morning. Yep, the howler monkeys have got you. Love these guys. They're delightful and they're everywhere. But I did prefer the white crested capuchin. They're right there. You can kind of see their white face. If you've seen Night at the Museum, it's a similar kind of monkey. Super playful, super energetic. There's a better picture that my friend Kaylana took of them. Cute little guys right there. Oh yeah, we went on a boat ride when we were in Palo Verde, along a river. We got to see a whole bunch of heron and egret and all sorts of crocodile. Here's one of those heron. I took this picture through my binoculars. If you're good at taking a picture through like a microscope lens, it's a pretty transferable skill in that regard. So I recommend trying that sometime. And this is probably the coolest thing anyone saw. This is not my picture. I got like the tiniest little glimpse of this thing moving. But inside of a dead tree when we were in Palo Verde was this margay. And what a neat little critter this was. They're very similar to ocelots. Yeah, like I said, I didn't really get any pictures of the toucans. I love the toucans, but yeah, just kind of use your imagination. They look something like this. That's the souvenir I got from my mom. Yeah. I care. I don't know. I'm not there to read the room. I don't know if anyone else cares. I care, though. So I promise you, I will do my best to be informative and get the point across, and I'll speed through this, don't worry. So, Project 1 was herpetofauna of various substrates. Now, what in the hell do those words mean? Essentially, we're looking at reptile and amphibian life across different material biomes. So we looked at the river, we looked at a disturbed substrate, which was sort of where human activity had occurred, and we looked at just the regular forest floor. And we did a survey, a time-based survey, of those areas, looking for different amphibian life and reptile life to sort of determine what the trends were. And despite our predictions, what we found was that at nighttime, the most variety could be found in the forest, which I believe was what we predicted. But we were not expecting for the most total sightings to be on the disturbed land, which was really interesting. There are factors that play into that, especially confirmation bias, because the flat open area is going to be easier to find the frogs and lizards. Whereas you'll see here, we didn't find many total sightings on the forest floor. Acacia ants. So, this was a really cool project. Essentially, acacia ants, pictured here, there are two types. I think that's Flavicornis, and that might be Paramyrilax? I don't know. So, the acacia tree has these thorns in it, which are expertly photographed here by Tomas. And the thorns are hollow. Ants will nest inside of them for protection. The tree also makes these little fruiting bodies for the ants to eat, and it secretes sap for them as well. Pretty cool deal. In response, the ants are meant to defend the tree. Our thought process was, well, a lot of these trees have a lot of damage from herbivores. They're missing leaves, they have damaged leaves, etc. So we wanted to see if the response time of the ants has a correlation to the health of the tree. And what we found was that there is some correlation. Now, this is the old version of the graph that doesn't have the color coding on the dots because I was still learning how to use JMP. The Spinicola ants, depending on the damage, there was more activity with less damage. And then with the Flavicornis ants, there was more ant activity proportional to more damage. That's a percentage there on the side. So, it kind of depends on the species of ant we learned. So there are both effective mutualists and ineffective mutualists, and that's kind of what we wrote our paper about when we did our research. It was probably the most interesting experiment we did of the bunch,